Grasslands stretch across continents with grass as the main source of food for herbivorous animals, yet most animals can't digest it. Deer, zebras, and elephants thrive here, but how? The secret is a remarkable alliance with microbes. These tiny partners break down cellulose, the tough fiber in grass, unlocking its nutrients. It's a story of two worlds, animals and microbes, working together to survive. Cellulose is the backbone of plant structure, made of tightly linked glucose molecules. Most animals, including humans, can't break these strong bonds. We lack the right enzymes. Microbes, however, possess the tools to unlock cellulose, turning it into usable energy. This partnership transforms an indigestible material into a vital food source for select mammals. Inside certain mammals, diverse microbes, bacteria, protozoa, fungi ferment cellulose. This fermentation produces volatile fatty acids, a crucial energy source for the host animal. The mammal provides a home, the microbes provide access to grass's hidden nutrition. Two main strategies evolved, Foregut and Hindgut fermentation, each with unique advantages. Cellulose gives plants their strength, forming tough interwoven fibers. Its beta-glycosidic bonds are nearly impossible for most animals to break. Without the right enzymes, cellulose passes through undigested unless microbes step in. Our digestive enzymes can break down starch, but not cellulose's tough bonds. Only certain mammals with microbial help can access the energy locked in grass. This enzymatic barrier defines who can turn a field of grass into a meal. In cellulose-digesting mammals, microbes produce cellulases to break tough bonds. Fermentation in oxygen-poor chambers releases volatile fatty acids, fueling the animal. Microbes themselves become food, adding protein to the diet. This symbiosis opens up grasslands as a food source, but not all mammals digest cellulose the same way. Ruminants, cows, sheep, deer, have a multi-chambered stomach for foregut fermentation. This system lets them efficiently extract energy from grass, thriving where others can't. Their four-chambered stomach, packed with microbes, is a marvel of evolution. It's a complex, efficient partnership between animal and microbe. The ruminant stomach has four chambers: rumen, reticulum, omasum, abomasum. The rumen is a vast microbial fermenter. The reticulum filters, the omasum absorbs water, the abomasum digests. Each chamber plays a unique role in breaking down tough plant fibers. Together they turn grass into energy and nutrients. Ruminants chew, regurgitate, and rechew their food, chewing the cud. This process maximizes cellulose breakdown, exposing more surface to microbial enzymes. The result, efficient extraction of nutrients from even the toughest grasses. Microbial fermentation produces volatile fatty acids, acetate, propionate, butyrate. These are absorbed and fuel the ruminant's body, supporting everything from movement to brain function. It's a perfect example of symbiotic efficiency. Microbes get food, animals get energy. Foregut fermentation is efficient but slow. Ruminants spend hours grazing and ruminating. Maintaining a complex stomach and microbial community is energy-intensive. Still, this strategy lets ruminants thrive on grasslands where others can't. But there's another way. Hindgut fermentation, used by horses, rabbits, and elephants. Hindgut fermenters, like horses and elephants, digest cellulose in the cecum and colon after the small intestine. Microbes here break down plant fibers, producing volatile fatty acids for energy. This method is faster, letting animals eat more and process lower-quality forage. It's less efficient than foregut fermentation, but offers speed and flexibility. Two strategies, one goal: unlocking the energy hidden in grass. The cecum and colon, often overlooked, are the fermentation hub in hindgut fermenters. The cecum acts as a fermentation vat, teeming with microbes that break down tough plant fibers. The colon supports this process, providing extra surface for microbial colonization and nutrient absorption. Horses have a larger cecum than rabbits, reflecting their grass-based diet. This system highlights the power of symbiosis, microbes unlocking nutrients for their animal hosts. Horses and rhinoceroses, those truly majestic herbivores that roam savannas and grasslands all across the globe, are prime examples of hindgut fermenters. Their large size and reliance on a diet primarily composed of grasses make them ideal candidates for this digestive strategy. Unlike ruminants, which spend a significant portion of their day chewing their cud, horses and rhinos can graze more or less continuously, with their digestive systems efficiently processing the steady stream of plant material. Their spacious hindguts, especially their capacious cecums, provide ample space for microbial fermentation to occur. Imagine these organs as vast fermentation chambers, working tirelessly to break down the tough cellulose fibers found in grasses. This continuous fermentation process allows horses and rhinos to extract a surprising amount of energy from their fibrous diet, fueling their active lifestyles. While hindgut fermentation may not be quite as efficient at extracting every last nutrient from plant material as foregut fermentation, it allows horses and rhinos to process large volumes of food quickly, compensating for any losses in efficiency. This strategy proves particularly advantageous in environments where food is abundant but of lower quality, such as grasslands with a high proportion of mature fibrous grasses. The success of horses and rhinos as hindgut fermenters really highlights the adaptability of the digestive system and its remarkable ability to evolve in response to ecological pressures. Their ability to thrive on a diet that would prove challenging for many other herbivores underscores the diversity of digestive strategies present in the animal kingdom and their crucial role in shaping the ecological relationships that define our planet. Rabbits, with their characteristic hopping gait and insatiable appetite for leafy greens, have taken hindgut fermentation to a whole new level. While their digestive system shares similarities with that of horses and rhinos, rabbits have evolved a unique adaptation known as checotrophy, or coprophagy, to maximize their nutrient extraction from their fibrous diet. This behavior, while seemingly unappealing, is a testament to the ingenuity of evolution and its ability to find elegant solutions to biological challenges. Checotrophy involves the consumption of specialized nutrient-rich fecal pellets known as checotrophs directly from the anus. These checotrophs, produced in the cecum, are coated in a mucus layer that protects their precious cargo of nutrients from the harsh environment of the stomach and small intestine. Imagine these pellets as time-release capsules delivering essential vitamins, minerals, and microbial proteins that would otherwise be lost in the digestive process. By consuming caecotrophs, rabbits essentially get a second chance at digesting their food. The nutrients within the caecotrophs, protected from digestion the first time around, are released in the small intestine, where they can be absorbed and utilized by the rabbit. This ingenious adaptation allows rabbits to thrive on a diet that would be nutritionally insufficient for many other herbivores. Caecotrophy highlights the remarkable diversity of digestive strategies present in the animal kingdom and the lengths to which evolution will go to extract every last bit of nutrition from a given food source. It's a reminder that nature is full of surprises and that what might seem strange or even repulsive at first glance often serves a crucial purpose in the grand tapestry of life. Hindgut fermentation offers speed and the ability to use lower quality forage, but with trade-offs. Because fermentation happens after the main site of nutrient absorption, Hindgut fermenters are less efficient than Foregut fermenters. It's a trade-off. Speed versus thoroughness, each with its own ecological advantages. Hindgut fermenters can quickly consume large amounts of food, thriving where forage is abundant but scattered. Whether it's a cow's multi-chambered stomach or a horse's large cecum, each adaptation is a unique solution to extracting energy from plants. The evolution of foregut and hindgut fermentation represents a fascinating example of how natural selection tailors digestive strategies to suit an animal's size, lifestyle, and ecological niche. Large herbivores, such as cows, elephants, and rhinoceroses, face a unique set of challenges. Their imposing size necessitates a large food intake to fuel their massive bodies, while their relatively slow movement and reliance on grazing as their primary mode of foraging limit their ability to be selective about their food choices. In these giants, hindgut fermentation has emerged as the dominant digestive strategy. Their spacious digestive tracts, particularly their capacious cecums and colons, act like giant fermentation vats, allowing for the continuous processing of large volumes of plant material. While hindgut fermentation may not be as efficient at extracting every last nutrient from cellulose as foregut fermentation, it compensates with speed, allowing these animals to process vast quantities of food quickly and meet their substantial energy requirements. Smaller herbivores, such as rabbits and rodents, face different pressures. Their smaller body size allows for greater agility and selectivity in their foraging habits. They can afford to be more selective about their food choices, opting for nutrient-rich plant parts or young, tender shoots. However, their smaller gut size limits their capacity for microbial fermentation. In these smaller herbivores, hindgut fermentation, often coupled with specialized adaptations like caecotrophy, provides a balance between speed and efficiency. The faster processing time of hindgut fermentation allows for a higher food intake, compensating for any losses in digestive efficiency, while caecotrophy allows for a second chance at nutrient extraction, maximizing the nutritional value of their diet. The type of fermentation system an animal has shapes what it eats and where it lives. Foregut fermenters thrive on tough grasses, while hindgut fermenters can eat a wider variety of plants. The plants animals eat also shape their gut microbes, leading to further specialization. This interplay between digestive system, gut microbiome, and diet shows how animals adapt to their environments, rainforest, grassland, or desert, through evolution. The ability of certain mammals to digest cellulose shapes entire ecosystems far beyond their own survival. Foregut and hindgut fermenters act as ecosystem engineers, transforming landscapes and cycling nutrients. Elephants, for example, topple trees in open woodlands, letting sunlight reach the ground and grasses flourish. This benefits grazers like zebras and gazelles, who depend on these grasses. Their dung returns nutrients to the soil, supporting decomposers and plant growth. The ripple effects of cellulose digestion connect herbivores, predators, and decomposers, shaping the web of life. From the sweeping plains of the Serengeti to the lush Amazon rainforest, the world's herbivores have evolved a remarkable array of adaptations to unlock the nutritional treasures hidden within plant life. We've journeyed through the intricate workings of two distinct digestive strategies: Foregut fermentation, exemplified by the ruminants with their multi-chambered stomachs, and Hindgut fermentation, employed by horses, rabbits, and a host of other herbivores relying on their capacious cecums and colons. These two pathways, while sharing the common thread of microbial symbiosis, highlight the remarkable diversity of solutions that evolution can produce in response to a common ecological challenge: the digestion of cellulose. Foregut fermentation, with its emphasis on thoroughness and efficiency, allows ruminants to thrive on a diet primarily composed of grasses, extracting maximum nutrition from even the most fibrous and nutrient-poor plant material. Hindgut fermentation, on the other hand, prioritizes speed and flexibility, enabling horses, rabbits, and their kin to consume a wider variety of plant material, from grasses to leaves to even some woody vegetation. This adaptability allows them to exploit a broader range of ecological niches, their digestive systems fine-tuned over millennia to suit their particular environments and dietary preferences. The digestive tracts of these remarkable herbivores, whether housing a rumen teeming with microbes or a capacious cecum humming with fermentative activity, stand as a testament to the power of natural selection to shape the anatomy and physiology of organisms to thrive in a world of diverse and often challenging environments. As we've delved into the intricacies of cellulose digestion, one overarching theme has emerged: the indispensable role of microbes. These microscopic marvels, often overlooked in our macroscopic world, are the true heroes of herbivory, the unsung maestros, orchestrating the breakdown of cellulose and the release of energy from a substance that would otherwise be inaccessible to most animals. Within the rumen of a cow or the cecum of a horse, a hidden world thrives, a complex and dynamic community of bacteria, protozoa, and fungi work tirelessly, their metabolic capabilities harnessed through millions of years of coevolution to break down the tough, fibrous cellulose molecules and release volatile fatty acids, the lifeblood of herbivores worldwide. This intricate symbiotic relationship, a delicate dance between host and microbe, is a testament to the interconnectedness of life on Earth. The herbivore provides the ideal environment for the microbes to flourish, a warm, moist, and nutrient-rich haven, while the microbes in return bestow upon their host the ability to thrive in a world dominated by plants. It's a reminder that the digestive capabilities of herbivores, their ability to shape ecosystems and influence the flow of energy through food webs, are ultimately dependent on the unseen world of microbes, the tiny but mighty engines that drive the grand ecological cycles of our planet. Our exploration of cellulose digestion, from the multi-chambered stomach of a cow to the cacotrophy of rabbits, has only scratched the surface of this fascinating and multifaceted topic. The world of herbivores, with their diverse digestive adaptations and intricate symbiotic relationships, offers endless avenues for further exploration and discovery. Delve deeper into the world of ruminants and discover the intricate interplay between diet, rumen microbiology, and milk production. Unravel the secrets of the equine digestive system and learn how horses have adapted to thrive on a diet primarily composed of grasses. explore the fascinating world of coprophagy in rabbits and other hindgut fermenters, and gain a newfound appreciation for the elegance and efficiency of this often misunderstood behavior. As we continue to unravel the mysteries of the digestive world, we gain a deeper understanding not only of the remarkable adaptations of herbivores, but also of the interconnectedness of life on Earth. The study of digestion, far from being a confined or esoteric field, opens a window into the fundamental ecological processes that shape our planet and sustain all life, including our own. So the next time you encounter a cow grazing peacefully in a meadow or a horse galloping across a field, take a moment to appreciate the unseen world within, the microbial marvels that make their herbivorous lifestyles possible. For within their guts lies a testament to the power of evolution, the ingenuity of nature, and the interconnectedness of all living things. Subscribe to Science World Explored for more animal biology, ecology, and fascinating digestive science.